Hello, my dear students. Today we are discussing the important questions that is from the sexual reproduction in flowering plants chapter. Already we have started the question answer discussion. So already we have discussed MCQ question in the last class and 2 mark questions, right? So already we have discussed these two sections. Today we are discussing the 3 mark questions and 5 mark questions. Okay. So you know that from this chapter, total 11 marks questions will be asked. Total 5 plus 3, 8 plus 2, 1 MCQ. Okay, so 1 or 2. It may vary. Like this, total 11 marks will be there for this chapter. So one of the very important chapter. So let us start with the 3 mark question. Okay, so this is the first question. Draw a label diagram of fertilized female gametophyte and mention the ploidy of any one of the products of double fertilization. Okay. Here, the diagram of the female gametophyte. Female gametophyte, that is nothing but a embryo sac. Okay. That is nothing but the embryo sac. You know the diagram of the embryo sac. So you should draw the embryo sac. Fertilized embryo sac. Okay. So here, synergids you should draw and zygote. Okay, so this is the zygote, and in the center you will find the PEN, that is primary endosperm nucleus. At the end you will find the antipodal cells. So these are the antipodal cells. So this diagram already I hope you have practiced antipodal cells. Okay, and mention the ploidy of any one of the products of double fertilization. Here, after the double fertilization, zygote will be formed. This ploidy of the zygote is, that is 2n, diploid. Okay, after the fertilization, after the double fertilization, PEN, primary endosperm nucleus will be formed, that is triploid in condition. Okay, after the fertilization, this is PN is triploid and zygote, that is diploid in condition. Okay, and this after the fertilization, the zygote that will develop into embryo. So these all are diploid in condition. Okay, so you have to mention the ploidy. So this is the diagram. Here, before the fertilization, ploidy also mentioned here, egg cell, that is a haploid, ploidy is n number of ploids, and before the fertilization. Okay, so this is before the fertilization. Synergids, they are also haploid, and the central cell. So central cell is already diploid. When it fertilizes with the male gamete, it will get triploid. Okay, then antipodal cells. Antipodal cells, they are located at the chalazal end and they have the ploidy n. They are also haploid. Then after the fertilization, these all are before the fertilization. These two after the fertilization. Zygote formed after the double fertilization and has ploidy diploid. Okay, zygote ಅಲ್ಲಿ ಏನಿರುತ್ತೆ ಯಾವಾಗಲೂ ಡಿಪ್ಲಾಯ್ಡ್ ಕಂಡೀಷನ್ ಇರುತ್ತೆ ಅಲ್ವಾ. ಎಂಡೋಸ್ಪರ್ಮ್ ಅಲ್ಲಿ ಏನಿರುತ್ತೆ ಆಲ್ರೆಡಿ ಡಿಪ್ಲಾಯ್ಡ್ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಇರುತ್ತೆ, ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಫ್ಯೂಸ್ ಆಗಿರೋದಕ್ಕೆ. ಸೊ ದ ಪ್ಲಾಯ್ಡಿ ಈಸ್ ಟ್ರಿಪ್ಲಾಯ್ಡ್ 3n. ಓಕೆ, ಬಿಫೋರ್ ದ ಫರ್ಟಿಲೈಸೇಷನ್ ಅಂಡ್ ಆಫ್ಟರ್ ದ ಫರ್ಟಿಲೈಸೇಷನ್ ಅನ್ನ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. Draw a LS of grass embryo diagram and label the following parts. Grass embryo, that is the monocot embryo ಅಲ್ವಾ. ಮೊನೊಕಾಟ್ ಎಂಬ್ರಿಯೋ ಡಯಾಗ್ರಾಮ್ ಅನ್ನ ಕೇಳಿದ್ದಾರೆ and label the following part: scutellum, coleoptile, shoot apex and radicle, root cap and coleorhiza. So these are the different parts you should label in the diagram. So practice the diagram of the monocot embryo. Grass embryo means that is monocot embryo. Okay, so here scutellum is there and shoot apex. So this is the shoot apex and epiblast, radicle. So this is the root cap, and coleorhiza, so that will develop into a root, and this will develop into a shoot. So this part develop into a shoot and this develop into a root. Okay. So this diagram is very, very important diagram. Sometimes this diagram will be asked for 5 marks with explanation. Okay. Next question. Draw a label diagram of transfer section of young anther. Okay. Here they are asking the transfer section of the young anther. So the young anther diagram, this is the young anther diagram and this is the enlarged part. Okay. So you should mention the uh, all the parts. So outer layer, this layer is the epidermis, and next layer, second layer is the endothelium layer, and after that, so sporogenous tissue. So these are the so next one is the middle layer. Okay, so this is the middle layer, this is middle layer, and this is sporogenous tissue, and these are the tapetum. Okay, so inside the sporogenous tissue they form the microspore mother cell, and formation of the pollen grains takes place inside the sporogenous tissue. This is the tapetum, and that nourishes the developing pollen grains. Okay, and that is having the more than one nucleus, tapetum. So this is the diagram of the young anther. ಈ diagram ಅನ್ನ ಬಿಡಿಸ್ಬೇಕು, and you should label and the layers, ಎಷ್ಟು ಲೇಯರ್ ಅನ್ನು ಬಿಡಿಸಿದ್ದಾರೆ ಅಲ್ವಾ, ಸೊ ಆ ಲೇಯರ್ ಅನ್ನ ಕರೆಕ್ಟ್ ಆಗಿ ನೀವು ಮೆನ್ಷನ್ ಮಾಡಬೇಕು. ಗೊತ್ತಾಯ್ತಾ? ಸೊ ದಟ್ ಈಸ್ ದ ಡಯಾಗ್ರಾಮ್. ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್, 5 ಮಾರ್ಕಿಗೂ ಕೇಳಬಹುದಾದಂತ ಕ್ವಶನ್. ಸೊ ದಿಸ್ ಕ್ವಶನ್ ಡಯಾಗ್ರಾಮ್ ಅನ್ನ 5 ಮಾರ್ಕಿಗೂ ಕೂಡ ಕೆಲವೊಂದ್ಸಲ ಕೇಳ್ತಾರೆ. ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಡಯಾಗ್ರಾಮ್. Next question. Explain briefly any 3 outbreeding devices to control the self-pollination. Plants, they develop the, they avoid the autogamy. Autogamy andre ಏನು? The pollen grains, they will transfer to the stigma of the same flower. Same pollen grains ಏನಾಗತ್ತೆ? Pollen grains ಏನಾಗತ್ತೆ? ಟ್ರಾನ್ಸ್ಫರ್ ಟು ದ ಸ್ಟಿಗ್ಮಾ ಆಫ್ ದ ಸೇಮ್ ಫ್ಲವರ್. ಒಂದೇ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಪಾಲಿನೇಷನ್ ಆದ್ರೆ ಅದನ್ನ ಅಟೋಗ್ಯಾಮಿ ಅಂತ ಕರೀತಾರೆ. ಟು ಅವಾಯ್ಡ್ ದ autogamous type of the pollination, they have developed the outbreeding devices. Means outbreeding devices andre enu? The name itself says that they develop the cross pollination technique. How the plants develop the cross pollination technique? ಇಲ್ಲಿ ನೋಡಿ, 3 ಅಂತ ಕೇಳಿದರೆ ಫಾರ್ 3 ಮಾರ್ಕ್ಸ್. ನೋಡಿ, I have mentioned all the types. In some species, pollen release and stigma receptivity are not synchronized. ಅಂದ್ರೆ ಕೆಲವೊಂದು ಸ್ಪೀಶೀಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರಿಲೀಸ್ ಆಗುತ್ತೆ, ಬಟ್ ಸ್ಟಿಗ್ಮಾ ಇಸ್ ನಾಟ್ ರೆಡಿ, ಸ್ಟಿಗ್ಮಾ ಇಸ್ ನಾಟ್ ರಿಸಿಪ್ಟಿವ್. ಓಕೆ, ಆರ್ the pollen stigma is ready but pollen grains will not release. So that is called as a that is not synchronized. Pollen grain release and stigma receptivity are not synchronized. Correct time ಅಲ್ಲಿ ಆದ್ರೆ ಸ್ಟಿಗ್ಮಾನು ರೆಡಿ ಇದೆ, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರೆಡಿ ಇದ್ರೆ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಪಾಲಿನೇಷನ್ ಆಗುತ್ತೆ. ಇಲ್ಲಿ ಸ್ಟಿಗ್ಮಾ, ಸೊ ದಟ್ ಈಸ್ ನಾಟ್ ಮೆಚೂರ್, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಮೆಚೂರ್ ಆಗ್ಬಿಟ್ಟಿದೆ, ಅಥವಾ ವೈಸ್ ವರ್ಸ. ಸೊ ಈ ತರ ಆದ್ರೆ, ಸೊ ದೇ ಆರ್ ನಾಟ್ ಸಿಂಕ್ರನೈಸ್ಡ್. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the pollen release. Okay, so that is also called as dichogamy ಅಂತ ಕೇಳ್ತಾರೆ. ಆ ಟೈಮಲ್ಲಿ ಏನಾಗುತ್ತೆ, pollen grains release ಆಗ್ಬಿಟ್ಟಿದೆ but stigma is not ready. Then the pollen grains transfer to the another plant stigma, another flower stigma ಅಲ್ವಾ, ಇನ್ನೊಂದು ಫ್ಲವರ್ ಸ್ಟಿಗ್ಮಾಕ್ಕೆ ಅದು ಟ್ರಾನ್ಸ್ಫರ್ ಆಗತ್ತೆ. ದಟ್ ಈಸ್ ಅ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್. ಅಕಸ್ಮಾತ್ ಆಗಿ ಸ್ಟಿಗ್ಮಾ ರೆಡಿ ಇದೆ ಬಟ್ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರೆಡಿ ಇಲ್ಲ ಅಂದ್ರೆ ದ ಸ್ಟಿಗ್ಮಾ ವಿಲ್ ರಿಸೀವ್ the pollen grains from the different plant. So that encourage the cross pollination. So second type nodi, in some other species the anther and stigma are placed at the different positions so that pollen cannot come in contact with the stigma of the same flower. Both these devices prevent the autogamy. ಇಲ್ಲಿ ನೋಡಿ, ಇದನ್ನ ಹಿಟಿರೋಸ್ಟೈಲಿ ಅಂತ ಕರೀತಾರೆ. ಈಗ ನೀವು ಹೈಬಿಸ್ಕಸ್ ಫ್ಲವರ್ ಅನ್ನ ನೋಡಿದಿರಾ ಅಲ್ವಾ, ಹೈಬಿಸ್ಕಸ್ ಫ್ಲವರ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ, ಸೊ ದಿಸ್ ಇಸ್ ಸ್ಟಿಗ್ಮಾ ಇಲ್ಲಿರುತ್ತೆ. ಸೊ ಜಸ್ಟ್ ಇಮ್ಯಾಜಿನ್ ದಿಸ್ ಇಸ್ ದ ಸ್ಟಿಗ್ಮಾ ಅಂಡ್ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಆರ್ ಹಿಯರ್ ಓನ್ಲಿ. ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರಿಲೀಸ್ ಆಗಿ ಸ್ಟಿಗ್ಮಾಕ್ಕೆ ಹೋಗಿ ರೀಚ್ ಆಗುತ್ತೆ. ಆದ್ರೆ ಇನ್ ಸಮ್ ಸ್ಪೀಶೀಸ್ ಸ್ಟಿಗ್ಮಾ ಈಸ್ in the different position and pollen grains are in the different position. Pollen grains ಬೇರೆ ಪೊಸಿಷನ್ ಅಲ್ಲಿ ಇರುತ್ತೆ, ಸ್ಟಿಗ್ಮಾ ಬೇರೆ ಪೊಸಿಷನ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಆಗೇನಾಗೋದಿಲ್ಲ, there is no contact, there is no pollination takes place, there is no contact between the stigma and the pollen grains. That is called as the heterostyle. So ಆಗ್ಲೂ ಕೂಡ ಏನಾಗುತ್ತೆ, there is a chance of cross pollination. The third device to prevent inbreeding is self incompatibility. ವಾಟ್ ಈಸ್ ಸೆಲ್ಫ್ ಇನ್ಕಂಪ್ಯಾಟಿಬಿಲಿಟಿ? ಇದನ್ನ ಕೇಳ್ತಾರೆ ಕ್ವಶನ್ ಅಲ್ಲಿ. ದಿಸ್ ಈಸ್ ಅ ಜೆನೆಟಿಕ್ ಮೆಕ್ಯಾನಿಸಮ್ ಅಂಡ್ ಪ್ರಿವೆಂಟ್ಸ್ ದ ಸೆಲ್ಫ್ ಪೋಲನ್ ಫ್ರಮ್ ದ ಸೇಮ್ ಫ್ಲವರ್ ಆರ್ ದ ಫ್ಲವರ್ ಆಫ್ ದ ಸೇಮ್ ಪ್ಲಾಂಟ್ ಫ್ರಮ್ ದ fertilizing the ovules by inhibiting the pollen germination or pollen tube growth in the pistil. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ, the pollen grain is not able to germinate on the self stigma. So ಈಗ pollen grains ಟ್ರಾನ್ಸ್ಫರ್ ಆಗ್ಬಿಟ್ಟಿದೆ ಟು ದ ಸ್ಟಿಗ್ಮಾ. ಈ ಸ್ಟಿಗ್ಮಾಕ್ಕೆ pollen grains ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಿದೆ, ಬಟ್ ದಿಸ್ pollen grain is not able to germinate. So that is not able to form the pollen tube growth on the pistil. So that is called as a self incompatibility. ಸೊ 3 ಮಾರ್ಕ್ಸ್ಗೆ ನಿಮಗೆ 3 ಕೇಳಿದ್ದಾರೆ. ಎನಿ 3 ಯು ಕ್ಯಾನ್ ರೈಟ್. ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ತುಂಬಾ ಸಲ 5 ಮಾರ್ಕ್ಸ್ ಕ್ವಶನ್ಗೂ ಕೂಡ ಕೇಳಿದ್ದಾರೆ. ಅನದರ್ ಡಿವೈಸ್ ಟು ಪ್ರಿವೆಂಟ್ ದ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಇಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ದಿ ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್. ಸೊ ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಸೊ ಓನ್ಲಿ male or female flower will be there. There is no presence of the both male and female reproductive structure in the same flower. Alva, if both male and female flowers are present on the same plant, such as castor and maize ಮತ್ತೆ castor ಏನಾಗಿರುತ್ತೆ, ಮೇಲ್ ಮತ್ತೆ ಫಿಮೇಲ್ ಫ್ಲವರ್ಸ್ ಒಂದೇ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಇಟ್ ಪ್ರಿವೆಂಟ್ಸ್ ದ ಅಟೋಗ್ಯಾಮಿ ಬಟ್ ನಾಟ್ ಗಿಟನೋಗ್ಯಾಮಿ. ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಒಂದೇ ಪ್ಲಾಂಟ್ ಇದೆ, ಬಟ್ ಮೇಲ್ ಫ್ಲವರ್ ಒಂದ್ ಕಡೆ ಇದೆ, ಫಿಮೇಲ್ ಫ್ಲವರ್ ಒಂದ್ ಕಡೆ ಇದೆ, ಬಟ್ ದರ್ ಈಸ್ ಎ ಚಾನ್ಸ್ ಆಫ್ ಗಿಟನೋಗ್ಯಾಮಿ ಅಲ್ವಾ, ನಾಟ್ ಅಟೋಗ್ಯಾಮಿ ಬಟ್ ಗಿಟನೋಗ್ಯಾಮಿ. ನೆಕ್ಸ್ಟ್ ನೋಡಿ, ಅನದರ್ ಟೈಪ್ ಈಸ್, ಸೊ ದರ್ ಇಸ್ ಎ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ದಿ unisexual flowers but in a different plants. Male flower is in one plant and female flower is in one plant. ಒಂದೇ ಪ್ಲಾಂಟ್ ಅಲ್ಲೂ ಕೂಡ ಏನಿರೋದಿಲ್ಲ, ಮೇಲ್ ಮತ್ತೆ ಫಿಮೇಲ್ ಫ್ಲವರ್ಸ್ ಇರೋದಿಲ್ಲ. ಸೊ they are present in the different plants. That is also encouraging the outbreeding devices. Okay, so here that is called dioecy. That is called in papaya. So you can observe this condition. So it prevents both autogamy and gitanogamy. Okay, so these are the different types of outbreeding devices. 3 anta kelidare for 3 marks. So you can write 5 for 5 marks. Okay, very important. Next question. Why bagging of emasculated flower essential during the hybridization experiment? Alva. So during the artificial hybridization, they will transfer the desired pollen grains to the stigma of the desired flower. Alva, desired plant flower. So ಅಲ್ಲಿ ಏನ್ ಮಾಡಿರ್ತಾರೆ, after emasculated flower means anthers are removed. Emasculation means anthers are removed. Anthers remove ಆಗಿದೆ, but stigma is there. But this stigma is bag. ಈ ಫ್ಲವರ್ ಅನ್ನ ಬ್ಯಾಗ್ ಮಾಡಿಟ್ಟಿದ್ದಾರೆ. ಬಿಕಾಸ್ ಸೊ ಬ್ಯಾಗಿಂಗ್ ಪ್ರಿವೆಂಟ್ಸ್ ದ contamination with the undesired pollen grains of the ಸ್ಟಿಗ್ಮಾ. ಸೊ ಇಟ್ ಪ್ರಿವೆಂಟ್ಸ್, ಈ ಬ್ಯಾಗಿಂಗ್ ಮಾಡೋದ್ರಿಂದ ಏನಾಗತ್ತೆ ಅಂದ್ರೆ, ಈಗ ಅನ್ಡಿಸೈರ್ಡ್ ಯಾವುದೋ ಬೇರೆ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಬಂದು ಪಾಲಿನೇಷನ್ ಆಗೋದನ್ನ ಅದು ಪ್ರಿವೆಂಟ್ ಮಾಡುತ್ತೆ ಅಲ್ವಾ. ಸೊ ದ ಬ್ಯಾಗಿಂಗ್ ಈಸ್ ವೆರಿ ಎಸೆನ್ಷಿಯಲ್. ಸೊ ಟು ಪ್ರಿವೆಂಟ್ ದ ಕಂಟಾಮಿನೇಷನ್ ಆಫ್ ಇಟ್ಸ್ ಸ್ಟಿಗ್ಮಾ ವಿತ್ ಅನ್ವಾಂಟೆಡ್ ಪೋಲನ್ ಜೊತೆ ಕಂಟಾಮಿನೇಷನ್ ಆಗೋದನ್ನ ಪ್ರಿವೆಂಟ್ ಮಾಡುತ್ತೆ ಅಲ್ವಾ. ಸೊ ಫಾರ್ 1 marks, ಮೆನ್ಷನ್ ದ ಸೆಲ್ಸ್ ಆಫ್ ಮೆಚೂರ್ ಪೋಲನ್ ಗ್ರೇನ್. ಮೆಚೂರ್ ಪೋಲನ್ ಗ್ರೇನ್ ಅಲ್ಲಿ ಯಾವ 2 ಸೆಲ್ಸ್ ಇರುತ್ತೆ, ವೆಜಿಟೇಟಿವ್ ಸೆಲ್ ಅಂಡ್ ಜನರೇಟಿವ್ ಸೆಲ್. vegetative cell and generative cell. Give the scientific name of plant that has viability record of 10,000 years. 10,000 year recording, viability recording ಇರೋ seed ಯಾವುದು? That is Lupinus arcticus. Okay. The scientific name is Lupinus arcticus. Draw a neat label diagram of megasporangium. Megasporangium is nothing but a ovule. Okay, ovule is called as a megasporangium. And in the megasporangium, so they will also ask like draw the neat label diagram of anatropous ovule diagram ಬಿಡಿಸಿ ಅಂತಾನು ಕೇಳ್ತಾರೆ. Anatropous ovule diagram ಬಿಡಿಸಿ ಅಂತಾನು ಕೇಳ್ತಾರೆ. This is the diagram of the anatropous ovule, megasporangium ಅಂತಾನು ಕರೀತಾರೆ. And this is very important diagram. You have to practice this. Okay. So mention the parts, hilum, funicle, and micropyle region. Then outer integuments and inner integuments. This is the nucellus and this is the embryo sac. ಸೊ ಈ ಎಲ್ಲ ಒಂದು ಪಾರ್ಟ್ಸ್ ಅನ್ನ ನೀವು ಲೇಬಲ್ ಮಾಡಬೇಕು, ಪ್ರಾಕ್ಟೀಸ್ ಮಾಡ್ಕೊಳ್ಬೇಕು. ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, ಅನಾಟ್ರೋಪಸ್ ಓವ್ಯೂಲ್ ಅಂದ್ರೂ ಕೂಡ ನೀವು ಈ ಡಯಾಗ್ರಾಮ್ ಅನ್ನ ಬಿಡಿಸ್ಬೇಕು. ದೆನ್ ಮೆಗಾಸ್ಪೊರಾಂಜಿಯಂ ಡಯಾಗ್ರಾಮ್ ಬಿಡಿಸಿ ಅಂದ್ರು ಯು ಕ್ಯಾನ್ ಡ್ರಾ ದಿಸ್ ಡಯಾಗ್ರಾಮ್. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್, ಡ್ರಾ ಅ ನೀಟ್ ಲೇಬಲ್ ಡಯಾಗ್ರಾಮ್ ಆಫ್ ಪೋಲನ್ ಗ್ರೇನ್. ಇದನ್ನ 2 ಮಾರ್ಕ್ ಕ್ವಶನ್ ಅಲ್ಲಿ ಲಾಸ್ಟ್ ಟೈಮ್ ನಾವು ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವಿ ಅಲ್ವಾ. ದ ಪೋಲನ್ ಗ್ರೇನ್ ಡಯಾಗ್ರಾಮ್ ದಟ್ ಈಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಮೆನ್ಷನ್ ದ ಪಾರ್ಟ್ಸ್, exine, intine and vegetative cell, generative cell. So these all parts you have to mention and practice the diagram which is given in your NCERT textbook. Okay. So ಪೋಲನ್ ಗ್ರೇನ್ ಡಯಾಗ್ರಾಮ್ ಇದನ್ನ 3 ಮಾರ್ಕಿಗೂ ಕೇಳ್ಬಹುದು, ಕೆಲವೊಂದ್ಸಲ 2 ಮಾರ್ಕಿಗೆ ಕೂಡ ಕೇಳ್ತಾರೆ. ಓಕೆನಾ. Next question. Discuss the modes of pollination in chasmogamous flowers. So chasmogamous flowers means which has the exposed stigma and style. Alba. So which have the long stigma and style. They are called as the chasmogamous flowers. And one more type is there. They are clistogamous flowers. Clistogamous flowers, they will not open at all. So ಅದು ಓಪನ್ ಆಗೋದಿಲ್ಲ. So the fertilization takes place. That is a type of the autogamy. ಸೊ ಅದು ಓಪನ್ ಆಗೋದಿಲ್ಲ. ಓಪನ್ ಆಗಿದ್ರೆ ಮಾತ್ರ ಅಲ್ಲಿ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಚಾನ್ಸ್ ಇದೆ ಅಲ್ವಾ. ಚಾಸ್ಮೋಗ್ಯಾಮಸ್ ಫ್ಲವರ್ಸ್ they are having the exposed stigma and style. And so here the 3 types of pollination in chasmogamous flowers are autogamy, also known as the self pollination, right? So here the self pollination also takes place, transfer of the pollen grains to the stigma of the same flower, and geitonogamy. So this geitonogamy also takes place in the chasmogamous flowers, means the transfer of the pollen grains to the same flowers of the same plant. Okay, same flower of the same plant, not same flower, flowers of the same plant, different flowers of the same plant. This is a type of cell pollination where the pollen grains are transferred from the anther to the stigma of the flower on the same plant. Xenogamy. Xeno means it is a type of the cross pollination. These 3 types can be observed in the chasmogamous flowers. When the pollen grains are transferred from the anther of one flower to the stigma of the flower of the different ಫ್ಲವರ್ ಆನ್ ದ ಡಿಫ್ರೆಂಟ್ ಪ್ಲಾಂಟ್. ಸೊ ಡಿಫ್ರೆಂಟ್ ಪ್ಲಾಂಟಿಗೆ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಿ ಅಲ್ಲಿ ಪಾಲಿನೇಷನ್ ಆಗೋದನ್ನ ಕ್ಸಿನೋಗ್ಯಾಮಿ ಅಂತ ಕರೀತಾರೆ. ಮೀನ್ಸ್ ಮೇಲ್ ಅಂಡ್ ಫಿಮೇಲ್ ಫ್ಲವರ್ಸ್ ಆರ್ ಇನ್ ದ ಡಿಫ್ರೆಂಟ್ ಪ್ಲಾಂಟ್, ಬೇರೆ ಬೇರೆ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಇರುತ್ತೆ ಅಲ್ವಾ. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್, ಡ್ರಾ ನೀಟ್ ಲೇಬಲ್ ಡಯಾಗ್ರಾಮ್ ಆಫ್ ದಿ ಎಲ್ಎಸ್ ಆಫ್ ದಿ ಫ್ಲವರ್. ಸೊ ದಿಸ್ ಫ್ಲವರ್ ಡಯಾಗ್ರಾಮ್ ಆಲ್ಸೋ ಆಸ್ಕ್, ಫ್ಲವರ್ ಡಯಾಗ್ರಾಮ್ ಅನ್ನ ಬಿಡಿಸ್ಬೇಕು. ಯು ಹ್ಯಾವ್ ಟು ಮೆನ್ಷನ್ ದ ಕೇಲಿಕ್ಸ್ ಅನ್ನ ಮೆನ್ಷನ್ ಮಾಡಬೇಕು, ಕೊರೋಲಾ ಮೆನ್ಷನ್ ಮಾಡಬೇಕು, ದೆನ್ ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟ್ ಅಂಡ್ ಫಿಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟ್. ಫಿಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಡಿಫ್ರೆಂಟ್ ಸ್ಟ್ರಕ್ಚರ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ ಫಿಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟ್ ಮತ್ತೆ ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಬೇರೆ ಬೇರೆ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಅನ್ನ ನೀವು ಮೆನ್ಷನ್ ಮಾಡಬೇಕು. ಅಂಡ್ ಸೊ ದ ಡಯಾಗ್ರಾಮ್ ಈಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. Clistogamous flowers invariably autogamous. Justify the statement. ನೋಡಿ, clistogamous flowers invariably they are autogamous. Why? Because clistogamous flowers which do not open at all. In such flowers, the anthers and stigma lie close to each other. They are lying close to each other. When the anthers dehisce in the flower buds, pollen grains come in contact with the stigma of and affect the pollination. Thus, clistogamous flowers are invariably autogamous as there is no chance of cross-pollination landing on the stigma. There is no chance of the cross-pollination because they do not open at all. So you should write the proper reason for this question. Why clistogamous flowers invariably autogamous? So this reason you should write, that is very important. Next question. How do flowers reward their insect pollinators? Explain. So 3-mark question. How do flowers reward their insect pollinators? You know that flowers some flowers they are pollinated by the insects. They have the attractive petal and attractive color, attractive fragrance to attract the insects. So how these insects are rewarded by the plants? How what the insects will get from the plants for their pollination? So here nectar and pollen grains are the usual floral rewards. When the insect which reach the flower, so it will suck the nectar which is present in the flower and the pollen grains. So already the pollen grains are released, that pollen grains, so they use as a food some pollen grains. So that is the floral rewards. That is the normal rewards. In some species, floral rewards are in providing the safe places to lay the eggs. So they provide the safe place to lay the eggs. At that process, the pollination will take place. For example, here 2 examples are mentioned. So you can note down. An example is that for tallest flower of Amorphophallus, the flower itself is about 6 feet in height. Okay, so in the Amorphophallus, so this type of the pollination takes place. And one more example is a similar relationship exists between the species of moth and the plant yucca, where the both species, moth and the plant, they cannot complete their life cycles without each other. Both are in the mutual relationship. Means both plant and the moth, the insects, they will benefited. So the moth deposits its eggs in the locule of the ovary. So it will deposit its eggs, and the flower in turn gets pollinated by the moth. The moth that will pollinate the flower, and moth it will lay the egg inside the locule. The larvae of the moth come out of the eggs as the seeds start developing. Okay, the larvae that will come out when the seeds start developing. So this is how the insects get reward from the flowers. Very important examples, also very important for your MCQ and 2-mark question. And this is the answer for the 3-mark question. Next question. Describe in brief different wall layers of anther with the help of neat label diagram. You know the 4 wall layers of the anther. 4 layers of the anther, that includes the epidermis, epidermis, endothecium, epidermis, endothecium, endothecium, tapetum, tapetum, and sporogenous tissue. And sporogenous tissue. These are the four wall layers. Already we know the diagram. With the help of neat label diagram ಅಂತ ಇದೆ. Diagram ಅನ್ನ ನೀವು ಬಿಡಿಸ್ತೀರಾ ಅಲ್ವಾ? ಯಂಗ್ ಆಂಥರ್ diagram ಅನ್ನ ನೀವು ಬಿಡಿಸ್ಬೇಕಾಗತ್ತೆ. ಇಲ್ಲಿ ನೋಡಿ. ಇಲ್ಲಿ and you have to explain. It is generally surrounded by four wall layers: epidermis, endothelium, middle layer and tapetum. Sporogenous tissue is present inside the tapetum. Okay, middle layer, I just mentioned, middle layer. The outer three wall layers perform the function of protection and help in the dehiscence of the anther and to release of the pollen. ಸೊ ಇಲ್ಲಿ ತ್ರೀ ಲೇಯರ್ಸ್ ಇದೆ ಅಲ್ವಾ, ಎಪಿಡರ್ಮಿಸ್, ಎಂಡೋಥೀಶಿಯಂ ಅಂಡ್ ಮಿಡಲ್ ಲೇಯರ್ಸ್ ಏನಿದೆ, ಸೊ ದಟ್ ಈಸ್ ಪರ್ಫಾರ್ಮಿಂಗ್ ದ ಫಂಕ್ಷನ್ ಆಫ್ ಪ್ರೊಟೆಕ್ಷನ್. ಸೊ ಅದು ಪ್ರೊಟೆಕ್ಟ್ ಮಾಡುತ್ತೆ. ದೆನ್ ಆಲ್ಸೋ ಹೆಲ್ಪ್ಫುಲ್ ಫಾರ್ ದ ಡಿಹೈಸೆನ್ಸ್ ಆಫ್ ದಿ ಆಂಥರ್. And innermost layer, tapetum, that is very important, that is helping for the that is providing the nutrition for the pollen grains. And cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. Next question. Differentiate between vegetative and generative cell. Differentiate between vegetative and generative cell ಅಂತ ಇದೆ 3 ಮಾರ್ಕ್ ಕೇಳುವಂತ ಪ್ರಶ್ನೆ. The vegetative cell is bigger, has abundant difference ಬರಿಬೇಕಾದ್ರೆ ನೀವು ಕಾಲಮ್ ಹಾಕಿ ಬರಿಬೇಕು, ಗೊತ್ತಾಯ್ತಾ? ಕಾಲಮ್ ಹಾಕಿ ಈ ರೀತಿಯಾಗಿ ಬರಿಬೇಕು. The vegetative cell is bigger, has abundant food reserve and large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with the dense cytoplasm and a nucleus. The generative cell divides mitotically to give rise to a two male gametes. Okay, so ಇದನ್ನ ಕಾಲಮ್ ಹಾಕಿ ಎರಡು ಡಿಫರೆನ್ಸ್ ಅನ್ನ ನೀವು ಬರಿಬೇಕು. Next question. Mention any 4 strategies adopted by flowering plants to promote the cell pollination. Cell pollination anna. So any 4 strategies adopted by flowering plants to promote the cell pollination. So you have to write the any 4 strategies that is adopted by a flowering plants to promote the cell pollination. Okay. Next question. Even though each pollen grain has 2 male gametes, why are at least 10 pollen grains, not 5 pollen grains are required to fertilize 10 eggs? ಈಗ ನೋಡಿ, embryo sac ಇದೆ ಇಲ್ಲಿ. So just imagine embryo sac ಇದೆ ಅಲ್ವಾ, ಇಲ್ಲಿ egg cell ಇದೆ. Just imagine, just egg cell is here. Here is one pollen grain. One pollen grain that releases the 2 male gametes. ಎರಡು ಮೇಲ್ gametes ಅನ್ನ ರಿಲೀಸ್ ಮಾಡುತ್ತೆ. Even though this 5 pollen grains are not enough to, why are at least 10 pollen grains, not 5 pollen grains are not enough to fertilize the 10 egg. ಈಗ 10 egg ಅನ್ನ ಫರ್ಟಿಲೈಸ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಎಷ್ಟು pollen grains ಬೇಕು? 5 ಸಾಕಾ? ಒಂದು pollen grain ಇಂದ ಎರಡು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ರಿಲೀಸ್ ಆಗತ್ತೆ. ಹಾಗಾದ್ರೆ 5 pollen grains ಸಾಕಾ? No, because one pollen grain that fuses with the egg and another pollen grain that fuses with the central cell. So that for each, so it is called as a double fertilization anta karitare alva. So for each egg cell, one male gamete is enough, but there is a central cell. So the another male gamete that fuses with the central cell. Adrinda, for one embryo sac, two male gametes are required. Alva, one embryo sac idre adike ಎರಡು male gametes ಬೇಕು. Alva, so the 10 pollen grains, so they are required to fertilize the 10 eggs. Define the seed dormancy. What is seed dormancy? So it is a state where seeds prevented from the germination. So they are prevented germinating even when environmental conditions are favorable. So that is called as a seed dormancy. This is a totally 3-mark question. Okay. Next we are discussing the important 5 marks question. Okay. So 5 marks question. First question is, explain the TS of young anther. So this already we have discussed in the 3-mark question. So this question, so they may be, sometimes it is asked in the 3-mark section or it may be in the 5-mark section. So young anther diagram already we have discussed in this session. Alva, ಏನಿರುತ್ತೆ epidermis, endothelium layer, middle layer and tapetum, sporogenous tissue. So ಇವೆಲ್ಲವನ್ನು ನೀವು ಬಿಡಿಸಿ ಲೇಬಲ್ ಮಾಡಬೇಕು. Read the question. The pollen grains are produced in the pollen sacs and they are represent the male gametophyte. Pollen grains represent the male gametophyte. In context to this, name the two cells of the pollen grains and mention the ploidy of the cells. Two cells are vegetative cell and the generative cell. Vegetative cell and the generative cell ಅಂತ ಇರುತ್ತೆ. They are haploid. Okay. ಈ pollen grains ಏನಿರುತ್ತೆ already haploid ಇರುತ್ತೆ. These cells also haploid. What type of the division takes place during the formation of pollen grains? Pollen grains formation ಆಗಬೇಕಾದ್ರೆ ಯಾವ ಒಂದು division ಆಗತ್ತೆ ಅಂತ. You know that first the microspore mother cell that is in a diploid condition ಅಲ್ವಾ ಈ microspore mother cell ಏನಿದೆ diploid condition ನಲ್ಲಿದೆ that will undergo the meiosis ಆಗಿ ಏನಾಗತ್ತೆ microspores ಅನ್ನ formation ಮಾಡುತ್ತೆ microspores. So they will develop into pollen grains alva. So these microspores are in a cluster of 4. They are called as microspore tetrad and microspores are released. So that will develop into a pollen grain. ಅದನ್ನ pollen grain ಅಂತ ಕರೀತಾರೆ ಅಲ್ವಾ. ಸೊ ಇಲ್ಲಿ ಮಿಯೋಸಿಸ್ ಆಗಿ ಡಿಪ್ಲಾಯ್ಡ್ ಇರೋದು ಹ್ಯಾಪ್ಲಾಯ್ಡ್ pollen grains ಅನ್ನ ಫಾರ್ಮೇಶನ್ ಮಾಡ್ಲಿಕ್ಕೆ ಏನಾಗಬೇಕು. ಸೊ ಇಟ್ ಶುಡ್ perform the meiosis. Next, how do you store the pollen grains for many years and mention its significance? Here, the pollen grains, they are stored in a liquid nitrogen for many years. So, that is in a minus 196 degrees Celsius. How do the pollen grains are stored for many years? That is stored in the liquid nitrogen. So, about minus 196 degrees Celsius temperature. What is autogamy? You know the definition of the autogamy? Transfer of the pollen grains, transfer of the pollen grains to the stigma of the same flower, stigma of the same flower. That is called as a autogamy. Explain the devices that the plants develop to prevent the autogamy. Autogamyಯನ್ನ ಪ್ರಿವೆಂಟ್ ಮಾಡಲಿಕ್ಕೆ breeding devices ಅಂತ ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ ಅಲ್ವಾ. Outbreeding devices. So it may be a self incompatibility ಇನ್ಕಾಂಪ್ಯಾಟಿಬಿಲಿಟಿ ಇರ್ಬೋದು, ಸೆಲ್ಫ್ ಇನ್ಕಂಪ್ಯಾಟಿಬಿಲಿಟಿ ಇರ್ಬೋದು, ಅಥವಾ ದೇ ಆರ್ the release of the pollen grains or maturation of the stigma is not synchronized, or they are at the stigma and pollen grains are at the different position. They are not able to reach the pollen grains are not able to reach the stigma. ಅದು ಕೂಡ ಆಗಿರ್ಬೋದು. Then formation of the uh, unisexual flowers, and the flowers that may be in a different plant. So these all are the techniques we have studied in the last question ಅಲ್ಲಿ ಡಿಸ್ಕಷನ್ ಮಾಡಿದ್ದೇವೆ ಅಲ್ವಾ. ಸೊ ಅದನ್ನೆಲ್ಲ 5 ಪಾಯಿಂಟ್ಸ್ ಇದೆ, ಅದನ್ನು ಕೂಡ ನೀವು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿ ಇಲ್ಲಿ ಬರಿಬೇಕು. ಸೊ ಫಾರ್ ದಿ 5 ಮಾರ್ಕ್ ಕ್ವಶನ್. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ನೋಡಿ. Explain the LS of grass embryo with neat label diagram. Already we have seen the diagram of this um, grass embryo, but you have to write the explanation also. Okay, so this is the diagram, very important. Here, embryos of monocotyledons possess only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards the lateral side of the embryonal axis. And its lower end, the embryonal axis, has a radicle and the root cap. Root cap is enclosed in an undifferentiated sheath that is called as a coleorhiza. The portion of embryonal axis above the level of attachment of the scutellum is called as epicotyle. You ನೋಡಿದಿರಲ್ವಾ epicotyle ಯಾವ ತರ ಇರುತ್ತೆ. And epicotyle has the shoot apex and few leaf primordia enclosed in a hollow folia structure that is called as a, so that will develop into a shoot. So this explanation you should write for 5 marks. And with the diagram, okay, diagram with explanation. Sometimes they will ask only diagram, or sometimes they will ask the explanation. So this explanation you have to write. What is double fertilization? Name the structures that are formed from the following parts after fertilization. After the fertilizer, first define the double fertilization. Double fertilization andre ಏನು? Double fertilization is a complex fertilization method where out of two sperms, one fuses with the egg cell and another fuses with the polar nuclei, result in a diploid zygote and triploid primary endosperm, so respectively. So this is totally called as a double fertilization. Here, see the question. Name the different structures that are formed from the following parts of after the fertilization. Ovary will develop into fruit. Ovary will develop into fruit. And PEN, primary endosperm nucleus, develop into endosperm that provide the nutrition for the developing embryo. Zygote develop into embryo. Zygote develop into embryo. Ovule develop into seed. Okay, for each 1 marks. So what is double fertilization? 1 mark. And for these remaining 4, 1 marks. Ovary develop into fruit. PN develop into endosperm. Zygote develop into embryo. Ovule develop into seed. That is for the 4 mark question. Next question. Sporogenesis, gametogenesis, and pollination are the important pre-fertilization processes in angiosperms. Explain the sequence of processes that occur after the compatible pollination. So, you know the pre-fertilization events and post-fertilization events, right? So, here pre-fertilization events are gametogenesis and sporogenesis, and the pollination. These are the pre-fertilization events. Here, they are asking to write the post-fertilization events. Okay. So, the 4 post-fertilization events are, so there is a formation of the endosperm. The primary endosperm cell divides repeatedly and forms the triploid endosperm tissue. The cells of this tissue filled with the reserve food materials and are used for the nutrition of the developing embryo. And the embryo develops at the micropylar end of the embryo sac where the zygote is situated. The zygote that will develop into embryo. Most zygotes divide only after the certain amount of the endosperm is formed. This is an adaptation to provide the assured nutrition to the developing embryo. Here, both in the monocotyledonous plant and the dicotyledonous plant, same type of the embryo development occurs, and the zygote give rise to the proembryo, globular embryo, heart-shaped embryo, and mature embryo. So like this, the mature embryo will develop. So this is also post-fertilization event. And seed. In angiosperms, the seed is the final product of the sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside the fruits, and a seed typically consists of a seed coat, cotyledon, and embryonal axis. Then fruits. The ovary develops into fruit, that is transformation of the ovules into seeds, and ovary into fruits proceeds simultaneously. So here these all are endosperm, then fruit development, seed development, and the embryo development. These all are the post fertilization events. It occur after the fertilization. Next question. Double fertilization is the unique feature of the angiosperms, and the products of this double fertilization is zygote and PEN, primary endosperm nucleus. In context of this, when a hexaploid plant pollinated by a tetraploid plant, find the ploidy of zygote and pen through the schematic illustration. Okay. So here, commonly, what happens? This is the normal. This is the egg cell, that is N, and this is already 2N condition, just, and these antipodal cells, they are in N condition, and this is also N haploid in condition. Okay. So just imagine this is a gamete. This is the female gamete, so it is having the haploid, and male gamete, that is also haploid. Okay, but it is formed by the diploid plant alva. So diploid plant ಇಂದ ಏನಾಗಿದೆ, haploid gametes formation ಆಗಿದೆ. ಈಗ they are asking here hexaploid plant ಅಂತ ಇದೆ. Hexaploid plant is pollinated by the tetraploid plant. Find out the ploidy of the here hexaploid plant andre, so that is a 6N condition, and tetraploid plant means that is a 4N condition. Okay, ಈಗ ನೋಡಿ hexaploid plant ಅಲ್ಲಿ ಏನಾಗಿದೆ, egg cell formation ಆಗಿದೆ, yes, here in the hexaploid plant is pollinated by the tetraploid plant. ಈಗ ಇಲ್ಲಿ ಗ್ಯಾಮೆಟ್ಸ್, ದಿಸ್ ಈಸ್ 6N ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಇದೆ. ಓಕೆ, ಹೆಕ್ಸಾಪ್ಲಾಯ್ಡ್ ಪ್ಲಾಂಟ್ ಅಂತ ಇದೆ. ಇಲ್ಲಿಂದ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಫಾರ್ಮೇಶನ್ ಆಗಿದೆ. ಸೊ ಹಿಯರ್ ದ egg cell, that is in a 3N condition, and this is also 3N condition, and here this is in a 3 plus 3, that is 6N condition, and these are 3N condition ಅಲ್ವಾ. ಈಗ ಏನಾಗ್ತಾ ಇದೆ, tetraploid they are, when they undergo the meiotic, they will produce the 2N male gametes. Okay, 2N male gametes ಏನಾಗ್ತಾ ಇದೆ, 2N male gametes fuses with the 3N zygote, sorry, 3N egg cell. ಆಗ ಏನಾಗ್ತಾ ಇದೆ, ಸೊ ಇಟ್ ಈಸ್ ಫಾರ್ಮಿಂಗ್ ದ 5n ಝೈಗೋಟ್ 5n ಆಯ್ತು. ಈಗ 6n central cell fuses with the 2n male gamete, that is in a 8n condition. So this is how the fertilization takes place in this plant. Okay, so ನೀವು ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, ಈಗ ಟೆಟ್ರಾಪ್ಲಾಯ್ಡ್ ಪ್ಲಾಂಟ್ ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. ಟೆಟ್ರಾಪ್ಲಾಯ್ಡ್ ಪ್ಲಾಂಟ್ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದೆ ಅಂದ್ರೆ, ಸೊ ದಟ್ ಈಸ್ ಇನ್ ಅ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಇನ್ ಅ ಡಿಪ್ಲಾಯ್ಡ್ ಕಂಡೀಷನ್. ಓಕೆ, ಈಗ ಹೆಕ್ಸಾಪ್ಲಾಯ್ಡ್ ಪ್ಲಾಂಟ್ 6n ಕಂಡೀಷನ್ ಅಲ್ಲಿರೋದು ಗ್ಯಾಮೆಟ್ಸ್ ಅನ್ನ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದೆ ಅಂದ್ರೆ ದಟ್ ಈಸ್ ಇನ್ ಅ 3n ಕಂಡೀಷನ್. ಓಕೆ, ಸೊ ದಿಸ್ ಈಸ್ ಹೌ ದ ಫರ್ಟಿಲೈಸೇಷನ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್. So this question is from the model question paper. Next question. So see the ovule, anatropous ovule. Which type of the ovule is shown here? That is the anatropous ovule. Okay. So this is nothing but a anatropous ovule. Anatropous ovule. And what is the function of the funicle? So this funicle that helps to attach the placenta. This is the funicle that helps to attach the placenta. That helps to attach the placenta. What are the protective layers of this tissue? So these are the protective layers of this tissue. They are called as the integument, outer integument and the inner integuments. Okay. Next question. Flowering plants have developed many devices to discourage the cell pollination and encourage the cross pollination. Comment. So this is nothing but this question is same as outbreeding devices. Outbreeding devices, what we have discussed, that you have to write here. Now 5 important points ಅನ್ನ ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವಲ್ಲ, ಅದನ್ನ ನೀವು ಆನ್ಸರ್ ಆಗಿ ಬರಿಬೇಕು. See the diagram. So this diagram is already you have seen in the NCERT textbook. Will this pollination confirm the fertilization? Yes, this is insect pollination, right? So insect pollination that confirms the fertilization. What are the floral rewards provided by the plants to the insects to revisit? Already this B question is already discussed, right? Already we have discussed that nectar and some they will provide the nectars and pollen grains as a reward. So other than this, they will provide some space to lay the eggs, and they will give the protection for the insects. Like that they will give the protection. So these all things we have discussed. Please refer that question. Okay, ನಾನು ಏನ್ ಕ್ವಶನ್ ಅನ್ನ ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ದೇನೆ ಅದನ್ನ ಇನ್ನೊಂದ್ಸಲ ರೆಫರ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಸೇಮ್ ಕ್ವಶನ್. Mention the characteristics of the flower in this pollination other than the floral rewards. So ಈಗ insect pollinated flower, ನಾವು ಲಾಸ್ಟ್ ಕ್ಲಾಸ್ ಅಲ್ಲಿ ವಾಟರ್ ಪಾಲಿನೇಟೆಡ್ ಫ್ಲವರ್ಸ್ ಅಂಡ್ ವಿಂಡ್ ಪಾಲಿನೇಟೆಡ್ ಫ್ಲವರ್ಸ್ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ಸ್ ಅನ್ನ ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ ಅಲ್ವಾ. ಈಗ ನೋಡಿ, ಇನ್ಸೆಕ್ಟ್ ಪಾಲಿನೇಟೆಡ್ ಫ್ಲವರ್ಸ್, majority of insect pollinated flowers are large, colorful, fragrant and rich in nectar. So insect ಅನ್ನ ಅಟ್ರಾಕ್ಟ್ ಮಾಡಬೇಕು ಅಲ್ವಾ, ಅದಕ್ಕೆ they are large, colorful, fragrant and rich in nectar. Nectar ಜಾಸ್ತಿ ಇರುತ್ತೆ. When the flowers are small, a number of flowers are clustered into an inflorescence to make the conspicuous. So ಇಲ್ಲಿ flowers ಚಿಕ್ಕದಾಗಿದ್ರೆ ಏನಾಗಿರುತ್ತೆ, they are in the form of inflorescence. Animals are attracted to flowers by color or fragrance. The flowers pollinated by flies or beetles secrete foul odors to attract these animals. So ಆ ಯಾವ ಒಂದು ಆರ್ಗಾನಿಸಮ್ಸ್ ಅನ್ನ, ಇನ್ಸೆಕ್ಟ್ಸ್ ಅನ್ನ ಅಟ್ರಾಕ್ಟ್ ಮಾಡ್ಬೇಕಲ್ವಾ, ಆ ಇನ್ಸೆಕ್ಟಿಗೆ ಅಟ್ರಾಕ್ಟ್ ಆಗುವಂತ ಕೆಲವೊಂದು ಫ್ರಾಗ್ರೆನ್ಸ್ ಅನ್ನ, ಓಡರ್ಸ್ ಅನ್ನ ರಿಲೀಸ್ ಮಾಡುತ್ತೆ. That is the characteristics of the insect pollinated flower. See the question. This is a model question paper question. So picture of mature angiosperm embryo sac is given below and answer the question follows. Which cell or nuclei of the embryo sac produce the zygote and primary endosperm nucleus? This egg cell, when it fertilizes with the male gamete, that produces the zygote, and this central cell, when it fertilizes with the male gamete, the central cell, when it fertilizes with the central cell or polar nuclei, central cell, when it fertilizes with the male gamete, it produces the primary endosperm nucleus. What is the ploidy of the antipodal cells? So these are the antipodal cells. They are in haploid condition. And primary endosperm nucleus. Primary endosperm nucleus, that is in a triploid condition. Okay. So this is 2 mark and this is a 2 mark question. Why the endosperm development precedes the embryo development? So this endosperm that provide the nutrition for the developing embryo. ಎಂಡೋಸ್ಪರ್ಮ್ ಏನ್ ಪ್ರೊವೈಡ್ ಮಾಡುತ್ತೆ, ದಟ್ ಈಸ್ ಪ್ರೊವೈಡಿಂಗ್ ದ ನ್ಯೂಟ್ರಿಷನ್. ಈಗ ಎಂಬ್ರಿಯೋ ಝೈಗೋಟ್ ಇಂದ ಎಂಬ್ರಿಯೋ ಆಗಿದೆ ಅಲ್ವಾ, ಈ ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ಮೆಂಟ್ ಗೆ ಇದು ನ್ಯೂಟ್ರಿಷನ್ ಅನ್ನ ಪ್ರೊವೈಡ್ ಮಾಡುತ್ತೆ. ಸೊ ದಟ್ ಇಟ್ ಪ್ರಿಸೀಡ್ಸ್ ದ ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ಮೆಂಟ್. ಎಕ್ಸ್ಪ್ಲೇನ್ ದ ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಅಲಾಂಗ್ ವಿತ್ ದ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಅಂತ ಇದೆ. ಸೊ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಡಯಾಗ್ರಾಮ್ ಅನ್ನ ನೀವು ಈ ರೀತಿಯಾಗಿ ಬಿಡಿಸ್ತೀರಾ ಅಲ್ವಾ. ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಅನ್ನ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು. ದ ಪೋಲನ್ ಗ್ರೇನ್ಸ್, ದ ಜನರೇಟಿವ್ ಸೆಲ್ ಆಫ್ ದಿ ಪೋಲನ್ ಗ್ರೇನ್ಸ್, ನಾನು ಜಸ್ಟ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೇನೆ. Pollen grain that releases the 2 male gametes when it reaches to the embryo sac. The 1 male gamete fuses with the egg cell and forms the zygote, and another male gamete that fuses with the central cell and forms the PEN. So this is called as a double fertilization. ಇದನ್ನ ನೀವೇನ್ ಮಾಡಬೇಕು, ಈ ಡಯಾಗ್ರಾಮ್ ಥ್ರೂ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು. ದಟ್ ಈಸ್ ಆಲ್ಸೋ ಅ 5 mark question. Explain the structure of mature microsporangium with neat label diagram. Mature microsporangium ಅಂದ್ರೆ ಹೇಳಿದ್ದೆ ಅಲ್ವಾ, ಓವ್ಯೂಲ್. ಓವ್ಯೂಲ್ ಅನ್ನ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು. ಯು ಹ್ಯಾವ್ ಟು ಎಕ್ಸ್ಪ್ಲೇನ್ ದ ಫ್ಯೂನಿಕಲ್ and hilum, micropylar region and chalajal region and integuments. They are the protective layers. ಇಂಟೆಗ್ಯುಮೆಂಟ್ಸ್ ಯಾವ ತರ ಇರುತ್ತೆ, ಇದನ್ನೆಲ್ಲ ನೀವು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು. ಓಕೆ, ಓವಿಲ್ ಡೈಗ್ರಾಮ್ ಅನ್ನ ಬಿಡಿಸಿ ಆ ಒಂದು ಎಕ್ಸ್ಪ್ಲೇನೇಷನ್ ಅನ್ನ ಮಾಡಬೇಕು. ದಟ್ ಈಸ್ ಫಾರ್ ದ 5 mark question. Describe the types of pollination based on the sources of pollen. Mention any 2 characteristics of wind pollinated flowers. ಕ್ವಶನ್ ಬಿ ಅನ್ನ ನಾವು ಲಾಸ್ಟ್ ಕ್ಲಾಸ್ ಅಲ್ಲಿ ಡಿಸ್ಕಷನ್ ಮಾಡಿದ್ದೇವೆ. ಪ್ಲೀಸ್ ರಿವಿಸಿಟ್ ದಟ್ ಕ್ವಶನ್. ಡಿಸ್ಕ್ರೈಬ್ ದ ಟೈಪ್ಸ್ ಆಫ್ ಪಾಲಿನೇಷನ್. ಇಲ್ಲಿ ನೀವು ಅಟೋಗ್ಯಾಮಿ ನೋಡ್ಬೋದು, ಹಾಗೆ ಗಿಟನೋಗ್ಯಾಮಿಯನ್ನ ಅಬ್ಸರ್ವ್ ಮಾಡ್ಬೋದು, ಹಾಗೆ ಝೀನೋಗ್ಯಾಮಿ. ಸೊ ದೀಸ್ ಆಲ್ ಆರ್ ದ ಟೈಪ್ಸ್ ಆಫ್ ದಿ ಪಾಲಿನೇಷನ್. ಇದನ್ನ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಬೇಕು. So students, we have discussed this much question in this session. That is very important questions, 5 marks question, and these all questions, they are from the old question paper or from the preparatory exams. Okay, please concentrate on these questions. So this will be helpful for your board exam. Thank you.